Hello, it's Rudena Nagar, and today we're going to be solving February-March 2021 Paper 1 Variant 2. Let's begin. Number 1. The photomicrograph shows onion cells. The actual length of the onion cell labeled Y is 350 micrometers. What is the magnification of the photomicrograph? So prior we've measured the image size and it's going to be 140 millimeters. And the equation for magnification is I am. In order to work out the magnification, we must divide the image size, that we know it's 140 millimeters, by the actual size, which is 350 micrometers. First of all, we have to convert the millimeters to micrometers by multiplying by 1,000. And then we divide by 350 micrometers. This gives us 400 magnification. So the answer is going to be D. Question number 2. Which row correctly shows the presence of cell structure in typical plant cells and in typical prokaryotic cells? Let's start with the first row. Centrioles, typical plant cells, and prokaryotic cells. First of all, centrioles are not present in plant cells. This is incorrect. For B, 70S ribosomes. Yes, they are present in both plant cells, in the chloroplasts and mitochondria, and in prokaryotic cells in the cytoplasm. So the answer is going to be B. Which letter identifies cell structures where semi-conservative replication of DNA occurs? First of all, DNA replication occurs in structures that actually contain DNA. In this case, chloroplast contains circular DNA. Same with mitochondria, also contains a circular DNA. So both of these are correct. For nuclei, it contains the normal linear DNA. In case of all of them containing DNA, so its replication is going to happen, and the answer is going to be A. Question number 4. Which parts of a cell contains ribosomes? So let's start with chloroplasts. Chloroplasts contain 70S ribosomes. Same with mitochondria. Golgi body do not contain ribosomes, and the cytoplasm contains 80S ribosomes, either free in the cytoplasm or in the rough endoplasmic reticulum, which is the site of protein synthesis. So 1, 2, and 4 is correct, and the answer is going to be C. Number 5. Which range of cell diameters is typical for prokaryotic cells? Here I've given you the range, and it's 0.1 to 5 micrometers size range. In this case, the only answer that fulfills this criteria is going to be B. Question number 6. Which row shows a combination of molecules that could be found together in a single isolated virus? So I've written a side note here. In theory, and beyond AS level, viruses contain all of these components. But DNA and RNA cannot be together in the same single virus, as written here in the question. Let's start with the most simple component present in a virus. In this case, is definitely going to be a protein coat. So we know that A and D are going to be correct. Now let's zoom in further into A and D. Here in A, both DNA and RNA are present in the same virus. This cannot be possible. It's either the DNA or the RNA. So A is going to be incorrect. Let's look at D. DNA is present in protein, so the only correct answer is going to be D. Number 7. A student carried out 4 biochemical tests on a sample of food. Which conclusion is supported by the results? Let's start with A. Fat is not present. Here we have an ethanol emulsion test, and if the solution turns cloudy, it actually means that fat is present. So A is going to be incorrect. B. Glucose is present. Let's look at Benedict's test. Here, Benedict's test is used to test for the presence of reducing sugars. In this case, the color changed from blue to yellow. This means that reducing sugar is present. In this case, it's telling us glucose. we cannot guarantee that that reducing sugar is going to be glucose because there are other reducing sugars. C, protein is present. Here we have the Biuret test and the solution turned from blue to purple. This means it's positive for protein presence, and the answer is going to be C. Let's go into D, starch is present. Here we have iodine solution. If starch was present, the solution would turn from yellow to blue-black color. In this case, it stayed yellow. Therefore, this means that starch is not present. Number 8. Which diagram represents a molecule of alpha glucose? Here I've given you the structure of alpha glucose, and by the way, this has to be memorized. Here we have this hydroxyl group at the bottom and both hydroxyl groups at the bottom. The hydroxyl group here is facing up, and the only answer that fulfills this criteria for alpha glucose is going to be A. Number 9. Which statements about glycosidic bonds are correct? 1. Condensation between glucose and fructose uses water and forms sucrose. Yes, glucose and fructose do form sucrose, but here it says uses water. Actually, condensation reaction releases a molecule of water, it doesn't use it. Using water is actually a hydrolysis reaction. Therefore, 1 is going to be incorrect. 2. Glycogen contains alpha 1 to 4 glycosidic bonds and alpha 1 to 6 glycosidic bonds formed by condensation. Yes, as we mentioned, glycogen is a branched macromolecule and is the storage polysaccharide found in animals. Since it is branched, it must have both 1 to 6 glycosidic bonds and 1 to 4 glycosidic bonds. In this case, this is correct. 3. The hydrolysis of amylopectin uses water to break glycosidic bonds and release alpha glucose. As we mentioned here above, that hydrolysis reactions uses water to break down the glycosidic bonds. Therefore, 3 is also correct. 4. Amylose contains beta 1 to 6 glycosidic bonds formed by condensation. First of all, amylose contains alpha 1 to 4 glycosidic bonds. It does not contain 1 to 6 at all because it is not branched. Amylopectin is what contains alpha 1 to 6 glycosidic bonds, so not even beta. Therefore, the correct answer is 2 and 3, and the answer is going to be C. It has blank bonds formed by joining fatty acids to another blank. Which row correctly completes this sentence? Let's start with A. Here it says a triglyceride has ester bonds. Ester is correct. Formed by joining fatty acids to glycine. Glycine is incorrect because glycine is an amino acid. In this case, a triglyceride is formed by joining a glycerol to 3 fatty acids by ester bonds, releasing 3 molecules of water. Let's move on to B. Here, hydrogen bonds, we know it's incorrect, and ester is correct. And 2, here we have glycerol, as we mentioned here. So the correct answer is going to be. The diagram shows a phospholipid molecule divided into 3 regions, X, Y, and Z. R. In region X represents a range of possible chemical groups. Regions X, Y, and Z affect the properties of cell surface membranes in different ways. Which row shows the effect of each region on the properties of a cell surface membrane? So let's start with the first column, and increases the permeability of a hydrophobic region. So what actually increases the permeability is unsaturated fatty acids. The reason for this is because the unsaturated fatty acids binds less closely than saturated, which actually decreases the permeability because they bind so close together, only if it's saturated. So here we're looking for unsaturated fatty acids. The only correct solution is therefore going to be Z. The second column repels polar molecules. So what repels the polar molecules is definitely going to be a hydrophobic or non-polar molecule. In this case, it's going to be Y and Z. And this cannot be X because X is the phosphate group. And as we know, phosphate is actually polar, so it attracts, it does not repel, so this cannot be correct. Next column, attracts water molecules. As we mentioned, because the phosphate group is actually polar, so it can form hydrogen bonds with water, so it attracts it. Therefore, the answer in this case is going to be D. 12. Which statement about collagen is correct? A. Tissues containing collagen fibers cannot stretch. This is incorrect because actually collagen has a high tensile strength and is able to stretch. B. Each polypeptide chain forms an alpha helix. Alpha helix is incorrect. Alpha helix is actually in the secondary structure of proteins. Actually, these polypeptide chains bind together in a triple helix. 3 polypeptide chains bind in a triple helix. Alpha helix is here just to confuse you. So this is incorrect. C. 3 helices are held together by hydrogen bonds. This is correct. The 3 helices are held together by mostly hydrogen bonds and some covalent bonds, providing the high tensile strength. D. 3 polypeptide chains form a single collagen fiber. 3 polypeptide chains do not form a single collagen fiber, they form a collagen molecule. Several of these collagen molecules bind parallel to each other, form the fiber. But in this case, fiber is incorrect. So the answer is going to be C. The chemical found in number of fruits. Catechol can be oxidized to a quinone by the enzyme catechol oxidase. So here catechol is the substrate and catechol oxidase is the enzyme. Catechol oxidase is inhibited by parahydroxybenzoic acid, which is structurally similar to catechol. In the presence of PHBA, the Michaelis-Menten constant Km for catechol oxidase increases. So here there are 2 hints that actually demonstrate that this is a competitive inhibitor. First of all, it said that it has a structurally similar structure to catechol, which means that it binds to the active site too, must be the same shape. Second of all, here it says the Michaelis-Menten constant for catechol oxidase increases. If the Michaelis-Menten constant increases, this means that the affinity of the enzyme to the substrate has decreased. This is only possible in case of a competitive inhibitor, because in a non-competitive inhibitor, the Km value is never ever altered. Let's complete the rest of the question. How does PHBA inhibit catechol oxidase? 1. PHBA and catechol can both bind to the enzyme but not at the same time. This is correct in case that this is a competitive inhibitor. 2. PHBA is a non-competitive inhibitor. This is incorrect and we've discussed the reasons why. Decreases the Vmax of the reaction. Okay, so in case of the Vmax decreasing, this is only possible if it's a non-competitive inhibitor, because in a competitive inhibitor, the Vmax is not altered. So this is incorrect, and the only correct answer is going to be B. Which statements about the fluid mosaic model of a membrane are correct? Fluid means that phospholipids and components are able to diffuse or move around. Mosaic is the scattered pattern of the proteins. Let's start with 1. The movement of phospholipids in the membrane is reduced if the fatty acid tails are saturated. We've discussed this in previous questions. As we all know, if fatty acids are saturated, they bind closer together, so decreasing the permeability. So this is correct. 2. Glycoproteins in the outer layer of the membrane can move. We've just said that it's a fluid mosaic, so fluid meaning that the components are able to move around. This is correct. 3. Channel proteins are fixed in position. This is incorrect because of the fluid point. So the only correct answer is therefore going to be B. Diagram 1 and diagram 2 shows the transverse section through a leaf changes when a leaf is moved from the solution X to solution Y, which row describes the water potential of the leaf cells and surrounding tissues in diagram 2 compared with diagram 1. Okay, water potential of leaf cells. Here we're talking about the leaf cells in diagram 2 compared with water potential of leaf cells in diagram 1. In diagram 2 here, we can see that the leaf is almost more inflated, meaning that water has actually entered the leaf, compared with solution X, where here it's obvious that the water has actually left the leaf because it's more curled. Therefore, water potential in diagram 2 is going to be higher. Here, a higher water potential means that it is less negative. So less negative is going to be correct. In the next column, water potential of solution Y compared with water potential of solution X. Here we're talking about the solution, not the leaf. Be careful. So water potential of solution Y, it must be higher. The reason for this is because the water goes down water potential gradient. Therefore, it is a higher water potential, so it's less negative because water went inside the leaf. So the answer is going to be A. 16. Which features must always be present for water to move between two solutions by osmosis? So osmosis is the movement of water down water potential gradient through a partially permeable membrane. So let's look at the suggestions that fit this description of osmosis. 1. Carrier proteins. Carrier proteins only must be present in facilitated diffusion and active transport. So has nothing to do with osmosis. So this is incorrect. 2. Cell surface membrane. Cell surface membrane in this case is not yet accurate, so let's just leave it till the end and cross out A and B because we know for sure that one is going to be incorrect. 3. Selectively permeable membrane. In this case, selectively permeable membrane, as we said, is correct for water potential gradient. Water potential gradient must be present because, as we said, that water moves down water potential gradient. So in this case, we know for sure that 3 and 4 is going to be correct. So the correct answer is going to be D. When pancreatic cells were placed in a solution of red stain called neutral red, the cytoplasm became red. The cells were then removed from the solution of neutral red. The red stain in the cytoplasm moved into vesicles which were exported from the cell, eventually leaving the cell colorless. So the dye went in the cell and both out of the cell. Now, which transport mechanisms could explain how this red stain entered and left the cell? Here, that it says that the living pancreatic cells were placed in a solution. This means that there's a high concentration gradient of red dye outside the cell. Therefore, it's going to move in the cell by normal diffusion. And here, it says that the red stain moved into vesicles. The only time the stain will go out in vesicles is during active transport or exocytosis. In this case, B is going to be the correct answer. 18. DNA molecules in a eukaryotic cell must be packaged compactly because their combined length is much greater than the diameter of the nucleus. Which structures are required during the process of packaging DNA molecules compactly? So, first of all, we know that DNA molecule is incredibly long, almost 2 meters long. So must be packed compactly to fit inside the 10 micrometer nucleus. The only way this is possible is when that DNA molecule is wrapped around something called histone proteins. Histone proteins is where the DNA molecules are wrapped around it, making it compact and able to fit inside the 10 micrometer nucleus. Therefore, the answer is going to be C. Which statements about mitosis are correct? 1. Mitosis results in genetically identical cells. This is correct. 2. Mitosis is involved in asexual reproduction. Yes, asexual reproduction is a feature of mitosis because it's a genetically identical cell too. 3. Mitosis occurs immediately after cytokinesis. This is incorrect because mitosis actually occurs before cytokinesis, where sister chromatids are moved to opposite poles of the cell. Therefore, the cells divide. So this is incorrect. 4. Mitosis can be used to repair damaged cells. Repairing damaged cells is incorrect. It repairs damaged tissues. The reason for this is a cell can't actually be repaired. A tissue can be repaired by providing more of that cell making up the tissue. In this case, the answer is going to be C. 20. A human chromosome is made up of different parts. Some parts of a typical human chromosome are more numerous than others, so in higher quantities than others. Which parts are listed in order from least numerous, so the least quantity, to the most numerous in a human white blood cell? Start with A. Centromere, chromatid, and histone proteins. As we all know, we have one centromere in a single chromosome. So here we have one. For chromatids, we have two chromatids in a single chromosome too. So two chromatids. Then histone proteins. We have millions of histone proteins in the making up the chromatids. Therefore, this seems to be correct. It's from the least to the most. B. DNA molecule. We all know that we have two DNA molecules in each chromosome. Telomeres. We have 4 telomeres at the end of each chromatid. So here are the 4 telomeres at the end of each chromatid. And for the centromeres, we have 1 centromere. So this is incorrect. For histone proteins, as we said, we have millions. For telomeres, we have 4. And DNA molecule is 2. This is also incorrect. So the only correct answer is therefore going to be A. A piece of double-stranded DNA containing 12 times 10 to the power of 3 nucleotides is transcribed and translated to produce a single polypeptide. What is the maximum number of amino acids in this polypeptide? Here it's saying a piece of double-stranded DNA. So one strand is going to have 6 times by 10 to the power of 3, because here 12 times by 10 to the power of 3 is for 2 strands. So as we all know, that 3 nucleotides code for 1 amino acid. So in order to find the maximum number of amino acids, we divide this by 3, the number of nucleotides, and the answer in this case is going to be C. And don't be confused about the part of the double-stranded DNA. 2. Which statements about complementary base pairing are correct? 1. Cytosine forms 2 hydrogen bonds with guanine. This is incorrect. Cytosine forms 3 hydrogen bonds with guanine. 2. Purines and pyrimidines are different sizes. This is correct because purines have double ring structures and pyrimidines has a single ring. So they are definitely different sizes, so this is correct. 3. Adenine forms the same number of hydrogen bonds with thymine as it does with uracil. This is correct. With both, it does form 2 hydrogen bonds. 4. The base pairs in a molecule of DNA are of equal length and equal width. Must be correct. Here it's talking about the base pairs. So in order to maintain the same distance of DNA or that shape of DNA, both of them must be equal length and width in able to form that DNA shape. Otherwise, it would look something like this. In this case, 4 is going to be correct and the answer is going to be D. 3. What are the products when a DNA molecule replicates? So, A. Two molecules of DNA, each made of a sequence of paired bases. So, when a DNA molecule replicates, let's just assume that this is the DNA, it forms two molecules of DNA by semi-conservative DNA replication. So, an old strand and a newly synthesized strand. So, two molecules is correct. Now, each made of a sequence of paired bases. And here it says paired nucleotides. When describing the structure of DNA, we actually use the monomers. In this case, the monomers of DNA is going to be nucleotides, not bases. So the answer is going to be B. And the reason that the two strands is incorrect is because, as we mentioned, it forms two molecules of DNA, meaning 4 strands, not 2 strands. So C and D is incorrect. 24. Which descriptions apply to phloem sieve tube elements and to xylem vessel elements? So as we all know, that xylem vessel elements is hollow tube, therefore meaning that it does not have any sort of cytoplasm. Contrasting phloem, as we all know that phloem has only a small amount of cytoplasm and it's called a peripheral cytoplasm. Therefore this is incorrect. 2. No end walls. This is only correct for xylem, but not for phloem. The reason for this is that phloem, we know that it has sieve tubes to maintain movement. So this is also incorrect. 3. No nucleus. Yes, this is correct for both because both do not have a nucleus. The only correct answer is therefore going to be D. 25. Some of the parts of a plant root involved in water transport are listed. Which sequence shows part of the pathway through which water can move across the root? So let's start with A, 2, 3, and 5. 2 is cell walls of the cortex cell. We can see the diagram here, so this is correct. 3, endodermal cells. Endodermal cells is correct. It passes through the endodermis. And 5, xylem vessels. After it passed through the cortex and the endodermis, it enters the xylem. So this is correct. Let's see the other suggestions and I'll explain why they are incorrect. 2, 1, and 5. So it's a common misconception, many people put 1. The reason for this is we know that the Casparian strip is actually on the endodermal cell, but the Casparian strip actually blocks the apoplast pathway, so not allowing the water to enter through it. So this is incorrect, and this was a common misconception. The only pathway that the water can pass through is through the endodermal cell, but not through the Casparian strip, because it's impermeable to water. So A is going to be correct. 6. What is an example of a mass flow system in a plant? A. The loading of amino acids from the surrounding tissues into companion cells. The loading of amino acids from the surrounding tissues is actually a process called active transport, where H+ ions or hydrogen ions are pumped out of the companion cell, and this requires ATP, so it's not mass flow. B. The movement of sap through the phloem sieve tube elements in the stem. This is correct because the movement of the sap is achieved down hydrostatic pressure gradient. And going down hydrostatic pressure gradient is going to be mass flow. So B is going to be the correct answer. The temperature of a tree trunk usually decreases slightly during the day. Which changes in the environmental factors during the day could cause the diameter to decrease even more? So, what causes the diameter to decrease is actually increased transpiration. The reason for this is when water or more water are removed through the stomata from the leaves, it creates a higher gradient, which pulls more water molecules upwards by either adhesion and cohesion. The reason why the diameter decreases with increased transpiration or more water leaving the leaf through the stomata is because cohesion and the movement of water upwards actually cause a tension and pulls the stem inwards due to the tension forces of cohesion. Therefore, here in the question, we're looking for the factors that increase the rate of transpiration. In this case, increased temperature, increase the rate of transpiration due to increased kinetic energy of water molecules. Then, increased humidity. Increased humidity lowers the water potential gradient in decreasing the transpiration. So this is incorrect. Increasing light intensity also increases the rate of transpiration. Therefore, the answer is going to be A. 28. Which statement about the role of co-transporter proteins in the movement of sucrose is correct? So, co-transporter proteins are found on companion cells, and when companion cells pumps hydrogen ions outside or on its cell wall, both H+ ions and sucrose go back together through that co-transporter protein into the companion cell. So both of them go into the companion cell together. So let's look at the suggestions. Co-transporter proteins pump hydrogen ions into the phloem sieve tube elements. This is incorrect. It's proton pumps that pumps hydrogen ions. B. Hydrogen ions diffuse through co-transporter proteins with sucrose. Yes, as we mentioned, this is going to be correct, and the answer is B. 29. Which row correctly describes the heart during atrial systole? Atrial systole is when the atria contracts. In this case, when the atria contracts, the wall of the ventricle is going to be relaxed because both of them can never contract at the same time. Atrioventricular valve. In the case of atrial systole, it's definitely going to be open. And the atrioventricular valves are present between the atria and the ventricles. The reason why they are open is because during atrial systole, there is enough pressure generated by the atrial walls able to push that atrioventricular valve open. Semilunar valve. This is where the semilunar valve are located. So, as we all know that during atrial systole is going to be ventricular diastole, meaning that the ventricle is relaxed. In this case, there isn't enough pressure to push that semilunar valve open. So in this case, it's going to be closed, and the answer is going to be D. The diagram shows a capillary, some tissue cells, and a lymph vessel. Which row correctly identifies arrows 1, 2, 3, and 4? Let's start with 1. 1, we could see something going from the red blood cell into the tissue cells. In this case, it could only be oxygen due to oxygen dissociation from red blood cells. Now, let's just skip 2 at the moment, and let's skip 3 at the moment. Let's go to 4, and it's going to be the second easiest. 4, we know that there's a lymph vessel. What goes into the lymph vessel is definitely going to be tissue fluid. So here we know that 1 is going to be incorrect. Same with 2, that's also going to be incorrect. Let's go to C. Here we have flow of water and small solutes compared with plasma proteins. First of all, plasma proteins are unable to go directly from to tissue cells. The reason for this is here it's not specifying. Large plasma proteins are too big to pass through the endothelial pores of the capillary. Therefore, plasma proteins are going to be incorrect. Same with 3 here. Here it says osmosis of water. This does not happen. And diffusion of carbon dioxide. This is correct because it's the waste metabolite of tissue cells. Therefore, the answer that seem to be correct is going to be C. The table shows the results used to draw an oxygen dissociation curve for adult hemoglobin. What is the percentage increase in the volume of oxygen per gram of hemoglobin when the partial pressure of oxygen increases from 3 kilopascals to 5 kilopascals? So the equation to calculate the percentage change or increase is going to be new value minus original value divided by the original times by 100. Implementing this in the question, here we have 3 kilopascals. This is the original value, and the 5 kilopascals is the new value. So what we do is 0.96 minus 0.58 divided by 0.58, because this is the original, times by 100. This is going to give us 65.5%, and the answer is going to be D. 32. The graph shows the oxygen dissociation curves for adult hemoglobin at 2 carbon dioxide concentrations. Which letters show measurements taken in the capillaries of the lungs and in the capillaries of active muscle? Here we have two lines, the line R to Q and S to P. It is very obvious that the line R to Q is going to be the line with the actual highest concentration of carbon dioxide. The reason for this, it's because it has the lower percentage saturation of hemoglobin with oxygen. Therefore, we know that the active muscle has the highest concentration of carbon dioxide and the least saturation of hemoglobin with oxygen because it uses up the most oxygen. Therefore, we know that R to Q is going to be the line of active muscle, and the answer is going to be R for active muscle. For the lungs, we know that line S to P is the line with the lower concentration of carbon dioxide. And we know that the lungs are the site of gas exchange, therefore meaning that it has the highest percentage saturation of hemoglobin with oxygen. In this case, for the lungs, it's going to be P. So R and P, and the answer is going to be A. 33. Which substances can combine with adult hemoglobin and reduce the oxygen carrying capacity of the blood? First of all, we have 1, carbon dioxide. Yes, it indirectly causes a reduce in oxygen carrying capacity of the blood. The reason for this is when carbon dioxide combines with water, it forms carbonic acid by the action of the enzyme carbonic anhydrase. This carbonic acid dissociates into hydrogen ions and bicarbonate. When hydrogen ions combine with red blood cells, they form hemoglobinic acid. And hemoglobinic acid has a lower affinity to oxygen, therefore oxygen is released. Therefore, it reduces the oxygen carrying capacity indirectly. 2. Carbon monoxide. Yes, carbon monoxide decreases the oxygen carrying capacity. The reason for this is both carbon monoxide and oxygen share the same binding site on hemoglobin. Second of all, hemoglobin has a higher affinity for carbon monoxide than oxygen, so carbon monoxide combines first. Therefore, this is correct. 3. Hydrogen carbonate ions. No, hydrogen carbonate ions do not decrease the oxygen carrying capacity. Is hydrogen ions, not bicarbonate. Hydrogen ions, as we said in the first point, that hydrogen ions combines with hemoglobin, forming hemoglobinic acid, and oxygen is released. So this is correct, and the answer is going to be B. 34. How many times must a molecule of carbon monoxide pass through a cell surface membrane as it diffuses from an airspace in an alveolus through cell in the capillary wall into a red blood cell? So let's just assume that this is an alveolus and this is a capillary wall made up of many capillary cells and a red blood cell. So as we all know that the alveolus is made of a layer, squamous epithelium. Therefore, for carbon monoxide to pass through the alveolus, it must pass the first membrane to enter the squamous epithelium cell, then into the second cell surface membrane to exit the squamous epithelium cells. So here in the alveolus, we have 2 membranes passed. Then the capillary wall. it also must enter the cell in the capillary wall, so passes through the first membrane and then exit the same cell of the capillary wall. So in the capillary wall, here we have two membranes also that has been passed. Then, in order to enter the red blood cell, it passes through a single membrane only to enter the red blood cell. Therefore, here in total, we have 5, and the answer is going to be C. The heart rate of a person at rest was recorded at intervals over 2 15-minute periods. During the first 5 minutes of the second 15-minute period, the person smoked a cigarette. The graph shows the results recorded during the 2 15-minute periods. What can be concluded from the graph? A. Carbon monoxide affects the heart rate. Here in the graph, we must use the data provided only, not the data we know. So, since carbon monoxide has not been mentioned, so this is incorrect. Same with B. Carbon monoxide also has not been mentioned anywhere in the graph. C. Nicotine increases the heart rate. Also, nicotine has not been mentioned anywhere in the graph, so this is incorrect. For D, the heart rate increases as a result of smoking. Yes, here we have the person smoked, and we could see that the heart rate starts to increase as the person starts to smoke. So this is the only assumption that we can make from this graph, and the answer is going to be D. 36. Emphysema is a type of chronic obstructive pulmonary disease. A person with emphysema has difficulties in breathing out. Why does emphysema cause difficulties in breathing out? To make things clear, emphysema and COPD has been removed from the syllabus starting from 2022. But we're going to cover it just in case. We know in emphysema, the walls of the alveoli collapse and the surface area decreases. So let's see the suggestions. A. Smooth muscle fibers in the airways are destroyed. No, it has nothing to do with the smooth muscles. B. Too little mucus is produced. This is also incorrect. C. The elasticity of alveoli decreases. This is correct, resulting in alveoli bursting and a decreased surface area. So, 37. Which factors contribute to outbreaks of measles after natural disasters? First of all, also measles has been removed from the syllabus starting from 2020. But anyways, measles, as we know, it is a virus. 1. Contamination of drinking water with untreated sewage. Contamination of drinking water is only a spread factor in cholera. So this is incorrect. 2. Lack of effective vaccination coverage. Yes, of course, if the people are not vaccinated, there is a common outbreak. People living in overcrowded accommodation. As we all know that measles is a virus, so it could be transferred by airborne droplets. So 2 and 3 incorrect is going to be 38. There are two main classes of antibiotics, broad spectrum and narrow spectrum. Broad spectrum antibiotics affect a wide range of bacteria, while narrow spectrum antibiotics affect the specific types of bacteria. Which uses of antibiotics may result in antibiotic-resistant bacteria? 1. A broad spectrum antibiotic given to reduce the effects of influenza virus. First of all, here we know the specific type of virus caused. And here we're using a broad spectrum antibiotic. We know that influenza is a virus. So if you use an antibiotic with a virus, it won't even kill the virus and would only cause antibiotic resistance. So this is correct. 2. A broad spectrum antibiotic to treat Mycobacterium tuberculosis. So Mycobacterium tuberculosis is a bacterium, but here we know the specific type of bacteria and we are using a broad spectrum. If we know the specific type of bacteria, we must use a narrow spectrum only to affect that bacterium. So in this case, antibiotic resistance will occur. 3. A narrow spectrum antibiotic to treat diarrhea and sickness in cattle. Here, diarrhea and sickness is only mentioned, so it is not really specific. In this case, if we do not know the specific type of pathogen causing the sickness, we must use a broad spectrum, not a narrow spectrum. So this is correct and will also cause antibiotic resistance. Therefore, the answer is going to be A. Now, people with autoimmune disease, myasthenia gravis, can have a treatment to relieve the symptoms. In this treatment, one of the components of their blood is removed. Which blood component is removed? First of all, we have to know the pathophysiology of myasthenia gravis. What happens is antibodies attack the neuromuscular junction. So, which component must be removed is the antibodies, because it cannot distinguish between self and non-self cells. So the answer is going to be A. Why are spleen cells fused with myeloma cells during monoclonal antibody production? So, myeloma cells are cancer cells, and they are fused with spleen cells. The reason for this is cancer cells are able to divide unlimitedly. So let's see the common suggestions. A. So the antibodies become specific for the necessary antigen. This is incorrect. B. So the resulting hybridoma cells can reproduce indefinitely. This also means divide unlimited number of times. So the answer is going to be B. Thank you very much for watching this video. If you think this channel is useful, I'd highly appreciate it if you would subscribe, share, and like this video.